next 10 minutes or so, I'd like to share with you some of the work we have been done in a group in the microfluidic areas. And we have been focusing on developing microfluidics for lab-on-chips, including sensor devices, sample processing techniques, material synthesis, and microreactors, and also microfluidic-based thermal management systems. And this is the work done largely with many government agencies, universities here locally and internationally, as well as the support from MCN through NF node of Victoria here. So I won't go through everything we did in the last few years from the group. I'll be briefly give you a few examples in chemical and biological sensing, sample precision techniques like micromixing separation, and also one example using droplet to do enzyme evolution. One microfluidic device is based on 4-channel plastic microfluidic chip. We were able to develop an integrated device, and we were able to demonstrate detecting chemical warfare agent. From the time you load the sample onto our chip, we can get a result within 30 seconds. For many of you might be aware, currently most of the commercial available devices, you need a few hours to get an analysis for some of the nerve agent we are interested in. So the other things I want to share with you that not only the fast speed, but also the high resolution. Some of the techniques people being used, you cannot resolve the fine details of mixed chemical in the solution. The other things, which is most important, I'd like to share with you that developing sensors is not just get it done in the laboratory. You need to demonstrate that the sensor can be worked in a real harsh environment. Demonstrate the sensor in the lab, just one step or probably 10 steps you need to achieve. I'll give you just one example that we have tested our chemical warfare agent device with some of the samples which is mixed with some of the interference chemicals. We test this one against 3 or 4 commercially available devices. I won't disclose which device we have used data for, but 3 of them used. You can see all of the devices we test is fooled by 3 ordinary household chemicals like deodorant and also petrols. Some of them even can be fooled by simple household detergent. And the false alarm rate can be high as 40%. So which means that if you feed the sensors with the chemical you design the device for, you can detect that. However, if you feed the device with a mixture of different things, that's a totally different story. So our device, we tested 66 of the interferent chemicals, and none of them show negative effect on our device. That's why we're really proud of the device we haven't done so far. So the second example I'd like to share with you is the lab-on-chip magnetic nanoparticle platform. As many of you probably know as well, magnetic particles have been used commonly in many of the biology labs due to the ease of to do biosample separation. We think we have gone a step further. We try to control the magnetic particles on microfluidic device to try to integrate the AC control of mixing separation in one single chip. We would like to bring down the analysis of few hours down to few tens of minutes, if not minutes. So this just shows one of the principles. This is just a top left corner, just showing the principle of the assay we're doing. So we're trying to develop a 100 virus. So we're using the virus surface G protein of the virus, attach that to the magnetic nanoparticles, use that as a capture mechanism to detect antibody in the host serum, which is infected with the 100 viruses. So we're using other fluorescent techs as a fluorescent technique for detection. We put the assays into microfluidic channel for detection. So what I want to share with you is that after a few thousand, even 10,000 dilution of the host serum, we can still detect the amount of antibody in the solution, which is indicating very high sensitivity detection of the virus or the antibody in the whole serum. And the whole assay at the moment we've done is roughly 1 hour. It's not integrated on the chip yet, but the assay we developed can get the thing done within an hour, even much faster than the current commercially available device used in many labs for similar work. The third piece of work I'd like to share with you is the work we're developing lab-on-a-chip device for evolving enzyme molecules. What we do here is using monodispersed, micron-sized water oil droplet as nanoreactors to perform biological chemical analysis, so by chemical synthesis. This is just a schematic of the functionality of the microchip we developed. It consists of droplet generation, which produces monodispersed droplet fusion. You add in reagent to the drop to perform assay. Detect the droplet to look for the chemicals you have synthesized in the droplet. Now lastly is how to sort the droplet. If you have a droplet of interest, you need to collect them. So this is just a video showing several functions, how the droplet assay works. And I'd like to see that on the bottom corner there is a signal, fluorescent signal detected from the droplet. At the moment, we're using quite a lot of DNA in the droplet. That's why it can produce many copies of the enzyme. In this case, it's OPDA enzyme. It's enzyme degrading organophosphate chemicals. Our next step, we would like to reduce the number of genes in the droplet to single copy. In that case, we were able to choose which droplet hold the desired gene. We do the second round of evolution. That's our ultimate goal. Having done protein synthesis in a droplet, the same platform can be used for doing many other things. For example, we use the droplet platform to synthesize peptide beads, to synthesize hydrogel beads, to synthesize engineered beads for cell applications. We also use the platform to synthesize highly porous nanoparticles. I want to show you one picture of the nanoparticle we synthesized in the platform. This is a high internal phase materials with a voltage of 6 to 70%, high surface area. You can imagine this kind of materials can be used for many applications such as drug delivery, catalyst, and enzyme applications. The next example I'd like to share with you is some of the mixing work we've done for microfluidic applications. This is using different techniques that Professor James Friend and Leslie Yard's group are doing. We use the sound to shake the fluids. The way we shake the fluids is utilizing the non-linear behavior of the interaction of sound with the fluids at the micron scale, which is called microstreaming. So we use a different frequency of the sound to induce different mixing patterns, to induce different streaming patterns. When different stream patterns work together in the same pot of liquid, they induce very violent shaking. And you can see here on top right corner is a mixing pattern of 100 micron channel. Without the sound, then liquid there is stationary. So we use the same principle just to demonstrate this technique can be used in conjunction with the commercially available device. We cut a small piece of 96 well plate, put onto our device. We can demonstrate our device can reduce the elapsed time by the order of 10, approximately 7 to 10 times, which is significant save in time. As many of you probably know, ELISA assay could be a very lengthy process from a few hours even to 1 day. With our mixing technology, it can be done 1 or 2 hours. This is the significant improvement of the current assay technique. The last one I want to share with you is the microfluidic device we have been developed for separating particles from fluids. In this case, we'd like to separate blood cells from blood serum. We use a combination of dielectrophoresis with the hydrodynamic force to push the particles or push the cells from the main channel to the side wall, so that the central part of the channel only contains fluids without any particles, and that's the principle. The way we did that, we're using dielectric ferrous force and hydrogen force, so that along the main channel can induce secondary flow, so that the particles can be pushed to the side without coming back. And the middle image is showing a few particles mixed together with the fluids. As you can see, the red and the blue color is a particle put on the channel, and it goes just sideways without being contained in the middle. The middle is the port I'd like to collect sample for. So this device has demonstrated we can separate particles up to 99.9% efficiency. We also use the device to separate blood cells, and also we can achieve 99% of efficiency. To summarize, we have developing technologies in lab-on-a-chip, in chemical biological sensing, in sample processing techniques for lab-on-a-chip applications. We develop techniques for material synthesis, and also microreactors for chemistry and biology. Of course, this supported work has been done by many of my students, postdocs, and staff in CSIRO as well, such as Florine, Yonggang, also outside collaborators. And I also would like to thank CSIRO, DSTO, Department of Prime Minister's Cabinet, Emergency Management Australian, Federal Police and Customs for supporting some of the work early on for the chemical sensor device. And also would like to thank Melbourne Centre for Nanofabrication for providing me the tech fellowship so that I can carry some of the work at the center. Thanks for your attention.